Welcome, everybody. I think it's time to get started here talking about the MDRs, a very, very exciting thing to talk about. You'll find that, once I get going, it's, kind of hard for me to hold myself back on this one because I just love it so, so much. Before I get, too far into this, I just want to say that this topic of the MDRs and how to become compliant with them, it covers it's kind of like a big topic. So, really becoming compliant with the MDRs is a little bit more about becoming compliant with the new ISO 10993-1 on biocompatibility for the first time or again, especially if you have grandfather devices. And these topics we could talk about for a really, really long time. Sometimes, my colleague Thor Rollins and I will get together. We can talk all day or for days. And in fact, we do that regularly. So Nielsen Labs holds 3-day seminars where it's just us rambling on and getting really worked up and excited. And if you want even more detail than what we can squeeze into 45 minutes, then you're welcome to think about coming to one of these seminars. I think there's a schedule there in the back. I think the next one is in Amsterdam, and then we have one in Philadelphia in the spring. So those are coming. Before I, get into the good stuff here with MDRs, let me just briefly introduce myself. So my name's Matt Jorgensen. I'm basically your standard nerd. I have a lot of academic background in chemistry. So I did a PhD in physical chemistry and did some fundamental research in physics of materials in Germany for a while before coming to Nielsen Labs. I've been at Nielsen Labs for a few years. By far the best place I've ever been. Great place to be. And one of the things I love being at Nielsen Labs is I get to be in this really sweet spot where I can, everybody wins. So I can help people save quite a lot of time and money by not doing a whole bunch of testing that they don't need, and get to kind of make a living doing that. And plus I get to come and see all of you nice people, which is exciting for me because I don't get out a lot. Okay, so what are we really talking about? So we're here to talk about the MDRs, and this is something that is really, really timely to be talking about, obviously, because I think we all know that the MDRs are coming, and they're coming at a time when there's been actually a lot of change on the regulatory landscape for medical devices. So in case you didn't know, there's a new ISO 10993-1 that just came out last fall, really, really fresh, that presents a fundamental shift in the way that we're thinking about biocompatibility. It's one that we've been talking about for a long time, someday we're going to be really using a risk-based approach. Well, now that's codified and embodied in this document, the 10993-1. And we also have the 18562, which covers biocompatibility for gas path devices. That's new. That's just a year and a half old. The 10993-18 is in the works. It's coming. That covers, the different way that we ought to do chemistry testing for medical devices. And the reason why this is all relevant, there's all of this stuff that's new. And then on top of that, we have the new MDRs. And this deadline, May of 2020, is, rapidly approaching. And the reason why this is relevant is because to become compliant with the new MDRs, you have to be compliant with the latest standards on biocompatibility. And these are all the new latest standards on biocompatibility. So all of these things are tied together in a beautiful web of regulatory stuff, right? So it's a good time to be thinking about it. There's so just in a nutshell, if you have a device on the market in Europe, then I think that that to have a CE marking, you have to be get your device approved by a notified body, and that what it means to be approved, for market in Europe is changing with these new regulations. So the way that this has been rolling out is in May 2017, the new MDRs were ratified. It gave a 3-year introduction period for people to read it and become comfortable with it. In May 2020, this is when MDR compliance is required for new devices or resubmissions. Now, what that means is that as you start to approach this May 2020 deadline, if you're before that, you can still, apply for CE marking under the previous rules, the MDVs. After that, you have to use the MDR. So it could be, if you recertify right before May 2020, there will be devices on the market in Europe for a while that aren't compliant with the MDRs. But then by the time you reach May 2025, really any device on the market in Europe should be MDR compliant. So so this is the timeline here. As you can see, we're approaching May 2020, and if you're used to the way things move inside like a big corporation with lots of products and lots of people that have to sign stuff, things can move kind of slowly. So May 2020 is very, very rapidly approaching, and it's not just manufacturers that are worried and like kind of scrambling to get everything in order for this deadline, but also the notified bodies themselves, verifying that products are compliant with the new MDRs is kind of burdensome for them too. So there's a lot of these that are just dropping out, and some that are saying, okay, we're getting so overwhelmed that we're not even going to take new customers. And so it's good to sort this out really right away. Okay, so some of the pinch points with the new MDRs, if I just take a whole bunch of pages of document and condense it down to 3 bullet points that seem to stick out to me, one is that there's no grandfathering provision. So what that means is that even if you've had a device on the market for a really, really long time in Europe and you've supported its safety because it's been on the market for a long time, that's not good enough to let it stay on the market, because they could say, well, the old rules weren't always perfect at protecting patient safety. So how do we know that when you first got it, or when this device first was on the market, that it was actually, safe or not? So if you can't grandfather, that means that when you resubmit for your CE marking, that you have to prove its biocompatibility as if it were new. And then there's an expanded definition of what is considered to be a medical device. So there's some things that have been on the market in Europe that weren't considered devices that now are. And then it also explicitly requires that a qualified person have a look at the entire, docket, and who is responsible for compliance. And when they say qualified, you need to be able to support that with something like a CV and some credentials, something that demonstrates that this person that's looking is an expert qualified to make that decision. So I think the no grandfathering part is probably the most significant thing, because it means we have folks that have huge catalogs of devices where maybe all of them or most of them have been grandfathered, and now they're like, holy cow, how are we going to take 200-300 medical devices and go through biocompatibility for all of them? And that's kind of a big deal. So that's what this is all about. So leading into that, we need to talk about how we're going to demonstrate the biocompatibility of these devices to be compliant for the MDRs. And one of the things that's really significant here is that now we have the new ISO 10993-1 that we need to be compliant with. And this document, the new 10993-1, so it was just released in August 2018. It replaces the 2009 version. Before that, it was a long time before there was any other version, and now suddenly there's a lot of changes happening. But I have to stop here and just plug this document and announce publicly how much I love it, because we finally have gone from something like that was codified as really a checkbox type of scenario where you're thinking about, okay, well, what tests do I have to order in order to demonstrate that our device is safe, to one where you're really encouraged to stop and think and know your device and know your materials and only test or evaluate where it's appropriate. So this is really sticks out, because in the previous version of this table, in the last version of 10993-1, these were all X's here instead of E's. But they replaced the X's with E's to just remind you with every single little box in that matrix that okay, we're not just Xing something off, we're evaluating, right? And evaluating means, that you might be able to evaluate not with just a test, right? It could be that you can evaluate by, understanding your materials really well, or you could be able to evaluate by borrowing some other data. And there's lots of ways to evaluate the safety of your device and these biological endpoints other than a test. So all of the X's have turned to E's, and at the header of the table, it used to be tests for consideration when you are evaluating a medical device. Now it says endpoints to be addressed in a biological risk assessment. So it's really clear that we're assessing this thing, not just testing. So while I love this, it does also require a little bit of like expertise to be able to go through this evaluation process. And I'll go through an example of how we do that here in a minute. So I guess what I'm saying there with the shift from X's to E's and all of this is that the 10993, the new 10993 is all about risk and evaluating risk and understanding what is the best way to mitigate those risks that you find. And this is also in line with the new FDA guidance as well. So this is the general flow that we go through when we're looking at evaluating the risk of a medical device to be compliant with the MDRs. You can see the first step here is it's a 3-step process. The first step is where we're making a biological evaluation plan. And this takes a look at a device in its totality and all the materials in the device and understands where the risks are. And it sets or prescribes a strategy to evaluate and mitigate those risks. Then we move into a testing and risk assessment phase where we may do some tests, we may do some written evaluations, we might do some chemistry and a toxicological risk assessment. And that's where we're specifically addressing all of those endpoints. And then finally, at the end, there needs to be a biological evaluation report. And this is a document that summarizes like everything that you did before. And these two things, like the bookends here, the biological evaluation plan and the biological evaluation report, are two documents that you may or may not have ever used in the past, but that are really expected now, and especially as you go to submit in Europe, is they want this report at the end. They're not just going to take a bunch of like test results. You need to have an expert that has looked at all of the tests and then makes a claim. Okay, this is the device. These are the endpoints that we needed to use. And, we tested or evaluated them and it's safe. Okay. So one of the things, as we go through this biological evaluation plan, one of the things that we do is we start by identifying the risks. And you have a nice guide for this right in 10993-1 where you can ask yourself some of these questions. And I encourage you to go through this flow chart later, where you're really trying to decide if you need to test. And really what you are doing here is it kind of has this approach where if you're going to test, you almost need to defend the fact that you had to test. So it's like, are you sure you need to test? Is the material the same as something that's already commercially available? Does the device have the same chemical composition and so on? And if you can't answer those questions, then you do have to test. But what it means is that you can't just test blindly, right? So you shouldn't. So if you test unnecessarily, you might need to be able to defend that. Okay, so all of this this risk analysis, the whole setup goes into this biological evaluation plan, and then we go into the evaluation stage. Finally, the evaluation, the biological evaluation report. So let's just step through this with a real example. And I pulled this from, I changed a couple of small things, but this is something that we've been working on right now over the past couple of months. So we have a medical device manufacturer that manufactures orthopedic implant devices. They have a whole bunch of devices. So they have a whole set. And, they're looking at this, I think in their catalog, they have something like 3,000 SKUs. All of those are like different parts that can be used together in a kit and so on. And one of these is this plate and screw set that you see here. And so this is like represents a particular, I guess, more simple application. So within the set, you could have 4 different plate sizes. There's 5 different screw sizes. The screws come color coded. They're like anodized to different colors. And then you have material options, either titania or stainless steel. Then, of course, they have plates and screws from one supplier, and then they have a backup supplier as well. So they're like, okay, we have all of these parts. We need to become compliant to the MDRs. Now, what do we need to do? And they, the engineers are like, well, okay, we have 36 line items here. If we think about how it can be used in a patient, that's like 80 different combinations. And they were getting ready to test like all different like 80 sets, trying to just make sure that they were covered overall. And then that's when they came to us, and I was like, whoa, 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 whoa. You know, like 80 times like a big biocompatibility program is really, really a lot of burden. Maybe there's another way that we can do this. So this is, you know, the way we go through this process and what we did. And I think this testing is still in progress now. So first, what we do is we take a look using 10993-1, what biological endpoints have to be addressed for this set of parts. And, you know, if you find yourself asking these questions, we have slide rulers in the back that kind of like lay this out for you. But this is a permanent contacting device in contact with tissue bone. So if you look in 10993-1 in that annex, this would be tissue bone C, and everywhere there's an E, that's a biological endpoint that needs to be evaluated. Then from this list of endpoints, we go through and we might ask ourselves, you know, what type of options do we have to address these endpoints? And we go through one by one. So from that table there on the left, this is a list of all the endpoints that have to be addressed one way or another. Even if you are sure that that endpoint's not a risk, you still need to like explicitly address it. So you could say, you know, look, we know stainless steel isn't carcinogenic. So it's not just, you know, enough to assume that everybody knows that. You have to say carcinogenicity is made out of stainless steel. It's proven to not be carcinogenic. So it's explicitly addressed. So, okay, what we do, we go down the list and we think of what can we do to address each of these. So the first one is cytotoxicity testing. Cytotoxicity, the endpoint. And here, you know, there are ways that you could think about, say, addressing this without running a cytotoxicity test, but our experience has been that regulators really do like to just see you conduct this test, even if you might be able to argue out of it. And they like it because it's a very, very sensitive biological test, and it's good to sort of screen or catch the things that you might not expect to be there. It's kind of like a catch-all for the unexpected. So in this case, we really recommend running a cytotoxicity test, even if you're really sure that it's going to pass. The next endpoint, sensitization and irritation, these are ones where you can either do the traditional biocompatibility testing, or you have a shot at doing a written evaluation to address those without doing the testing. There's kind of pluses and minuses to each, but what we have found is that chemistry testing is usually not, while as a chemist I feel very strongly that I could support that scientifically, it's usually not very well accepted by regulators. So we would generally only go with a written assessment or traditional testing for that. The material mediated pyrogenicity is one that for now is really best to do just the testing. For the systemic toxicities, acute, systemic, subacute, subchronic, chronic, genotox, and carcinogenicity, you really could do any of those three things to address those endpoints. You could do, and we do this all the time, just a straight pen and paper exercise where you say, we know that this device isn't going to cause these effects because of X, Y, and Z, and this can be really convincing pen and paper. Well, of course, you can always do the traditional biocompatibility testing for those, but these are really burdensome tests. A lot of them are very long and expensive, so you want to try to avoid that if at all possible. Or you can do chemistry testing followed by a written evaluation. So what we often find ourselves trying to choose between is the top one, a written evaluation without testing, or a written evaluation with chemistry testing. And it really depends on what data the sponsor has on hand that we can use to aid in this evaluation. And then finally, for implantation, you really have two options there, either a written evaluation or to do the traditional biocompatibility test. The reason why chemistry doesn't work for this one is that implantation testing really evaluates the local interaction of tissues with the surface of the device. So it's kind of like a surface texture thing as much as anything else. And that's just not something that you're measuring with these traditional chemistry tests. Okay, so after going through all of these biological endpoints for our implant set, we are left with, essentially two possible strategies that we presented to the customer. We said, okay, look, we can do this way or that way, depending on how good the data is that you can give us. And so one option, if they had no usable existing data, so if they're like, yeah, we know it's stainless steel, but we don't know what grade of stainless steel, and we don't know how it's machined and all of that stuff, then this would be more of the type of plan that we would have. So for the big three, cytotoxicity, sensitization, irritation, and MMP, we would do the traditional biological testing. For all the systemic toxicities, we would do chemistry testing and a toxicological risk evaluation. And then for the implantation test, we would need to run that. And again, we haven't yet decided, which of all of those parts we're going to sample. We're just like thinking of the strategy of, how we're going to address endpoints. On the other hand, if they have really good information, lots of usable data on hand, then this can really limit the amount of testing that needs to be done. So if they had lots of data and we were kind of like encouraging them to like dig through what they've been given from their suppliers and stuff, then we still would run cytotoxicity testing, but sensitization and irritation, we could do a written assessment for that. MMP, they would still be doing probably as part of their sterilization plan anyways. And then for all of the others, we could make a written assessment based off of the data that they have. Now, the case that I'm showing here, it's a little bit special because I chose an orthopedic implant device that's made out of a well-known, well-characterized material like, CP titanium or stainless steel. Because with those materials, we know what the composition of the material very, very well. And if the implant's finished according to a certain ASTM specification, we also know what the surface is like. And then under those conditions, we can really say a whole lot without testing, so long as the supplier or the manufacturer can really prove that the device is free of manufacturing residuals. So it's a good plug for a cleaning validation. So if you can really prove that your device is clean and you have a device made from a really well-known material, then that can help you a whole lot in avoiding unnecessary testing. But if your device is made out of a plastic material, if we're talking about like a catheter set or some tubing set, even if it's clean, you probably are still going to need to do some testing, just because plastics are so ambiguous as to what they can release to the body. So in this case, they did have really great cleaning validation data, so we were able to limit their testing quite a lot just to the cytotoxicity and the material-mediated pyrogenicity. Of course, we have to review tons of cleaning validation data, but that's a lot less burdensome than running through the test. Okay, once you have the evaluation strategy figured out, then it comes to family groupings. And family groupings is where you're going to take a subset of this catalog of parts that you have, and you're going to pick certain pieces and argue or make a written argument why those pieces that you picked apply to everything in the catalog. And this has to all be well written and traceable, so that if an auditor came to your factory and is like walking through the warehouse and points to some random screw and says, okay, show me how you know that screw is safe, that you can say, okay, well, this screw has this part number. We showed that it was equivalent to this other screw, and we tested that screw, and that screw was safe, that type of thing. So within this set, we had 4 different plate sizes, 5 different screws, 2 different material types, and so on. So what we did is we said, okay, well, let's try to think of the worst case construct for this, and that's what we'll test. And we'll have a nice table here where we say, okay, these parts apply to all of these other ones. So we took the largest plate size and the largest screw size, and we said, okay, that plate and that screw is representative of all the other plates and screws. We wanted to test the titanium and the stainless steel separately because they came from different manufacturers or suppliers. We know that these different environments and manufacturing processes can impact the safety. Then we pooled the plates of the same material, even if they have the same processing. And then we said that the colored screws, if this is made by anodization, then if the coloration is by anodization, then the coloration doesn't really impact the safety. So we said, okay, the color doesn't matter. And then we took 1 or 2 sets of parts, and this can represent the entire collection that way. And it's all written nicely in a table. So in the end, since they tested the titanium and the stainless steel separately, we're really looking at two sets, and that's it to represent this entire family of parts. Okay, so then the tail end of this, so this goes into the testing and risk assessment phase. So we made a plan. We looked at what endpoints we needed to evaluate. Then for those endpoints, we devised a strategy. We said, okay, we're going to look at the cleaning validation. We're going to run a CITO and the MMP. And then we start into the testing phase. So in this testing phase, here it says, testing and risk assessments. So one of the things that we're doing here with the risk assessment is we're going to say, let's take a look at this cleaning validation data and try to address these biological endpoints. And we do this in the form of a written document that they can say, okay, well, how do you know this isn't a sensitizer? We say, oh, well, here in this document, we show, for example, that 316L has a really, really long history of being, used in medical devices. We at Nielsen Labs have, tested 316L stainless steel thousands of times. It's probably on test right now in replicate. And so we don't really need to test this material again to know that it's not a sensitizer. Everybody knows that. And if the device is clean, then we know that there couldn't be any sensitizers left on there. And so that's why sensitization isn't an endpoint that's concerning. And we do this in a biological risk assessment in written form that any auditor could review. Another type of assessment that we do, now it gets a little bit different from the orthopedic device example because we didn't have to do chemistry testing on that one. But in cases where we do the chemistry testing, then we do a toxicological risk assessment. This is where we take the data of extractables and leachables chemistry testing and interpret what the toxicological risk would be. So, the toxicologist will make like a specific claim about carcinogenicity and genotoxicity and so on based off of these results. Now, the reason why I want to bring this up here, and I'm going to talk about chemistry a little bit just because I can't hold myself back. But like as you go through this process of becoming compliant with the MDRs, it's pretty likely, you could have some cases where you have very minimal testing, but more often than not, you're going to be in the case where you have limited data available and you'll have to do chemistry testing and a toxicological risk assessment. It's actually pretty likely that happens. If you have a device that has plastics, which many devices do, then you're definitely in that category, or if you don't have good cleaning validations. So when you find yourself in this situation, you have to do chemistry testing. It's good to start the process by thinking about the toxicological risk assessment that you're going to have to do. So it would be good to know, who is your toxicologist going to be? Is it somebody inside your own organization? Is it somebody that you're hiring to assess the data? And you should start the conversation with them before you even order the chemistry testing, because I know a few toxicologists, and these folks tend to be very, very particular about the data that they're accepting. And in the end, they have to sign their name to this assessment. And if the data doesn't match what they want, they may just say, sorry, I won't do it. Do the testing again. So it's good to start with the toxicologist, understand what claims they're going to make in a toxicological risk assessment, and ask them what type of data that they want in order to support this. And that's the way that we are approaching chemistry testing at Nielsen Labs. We start with the toxicologist, we understand what they need, and then we try to deliver that data. Okay, and so when we start this chemistry testing, and this is informed by our own toxicologist, we really start this in a way where our toxicologist, and this is in line with the standards, that the new 10993-18, we really want to cast a wide net. And the example that I usually give is with this 3D printed spa can. So this is a real medical device. It's intended to be implanted in the body next to the heart so that you can keep the memory of Leonard Nimoy with you always. And if you were the Star Trek fan that was going to receive this implant, and if you were the toxicologist that were going to evaluate it, would you say, well, I think we should only test for compounds that are on this short list of chemicals of concern, that the CMRs in Europe? Or would you say, well, I know that there can be crazy like photoactivators in 3D printed stuff, let's just screen for those? No. You would say, okay, look, we ought to look for photo activators and things on the CMR list, but we also need to look for things that we don't expect to be there. Weird oil dripping from the ceiling onto the assembly line or like whatever. So in the end, you cast a really wide net and you look for anything and everything that could be in or on the device. And the way that we do that, so if you think about anything that could be in or on a device from like a chemistry perspective, in chemistry, we often divide all substances into two groups. We think about organic and inorganic. And then on the inorganic side, we can kind of lump those all together and address those as one type of test. And then on the organic side, we can subdivide this into a few different groups. We tend to do this in terms of like volatile organics and then semi-volatile organics in a different group and non-volatile organics. Now, when you go through on the organic side, as you can imagine, if we're talking about we want to look for anything, that means that we're screening for thousands and thousands and thousands of compounds, right? So we have to use a suite of analytical methods to try to do that. And those methods, if you're going to order chemistry for a toxicological risk assessment, if those methods don't include chromatography, then you should run away, right? Because this task can only be accomplished if chromatography is part of it. If they say, oh, we're going to do a non-volatile residual and look at the residue by FTIR, it's terrible, right? These compounds have to be separated from each other if they're going to be identified. You get like this soup of sludge from the medical device, and you got to like separate everything to do it. So chromatography is something that's required. It has to be there. It separates all the different compounds from each other. Once they're separated, they have to be identified. And it really matters who you go to and what the nitty gritty details are, like how good this identification is. So that's one of these things where, at Nielsen Labs, we've been, testing medical device materials and pharmaceutical materials for a very, very long time. And we keep track of all the compounds that we find. And so the compounds that we have experience with, we've built up into our own internal database, and we rely on that a lot. But if you don't have this big internal database, then what you use is something like the Nist database, which we use too, right? Because we don't have everything. And the Nist database is 200,000 some odd compounds that you can screen against. And then you have to identify these using mass spec. So that's internal database and Nist and Wiley. You take everything that you can to identify as many as you can. So that's exactly what we do. On the organic side, for small molecules and medium-sized molecules, we use a couple different flavors of gas chromatography with mass spectroscopy. For big molecules, this is where like really the art of mass spec comes into play, and where this private or internal database becomes really important. We use liquid chromatography. And then for metals and metal oxides, we use an inductively coupled plasma method, either ICP OES, so that's optical emission spectroscopy, or ICP MS, that's mass spectroscopy. And using this suite of methods, we can screen for just about everything and identify them, measure the amount, the concentration, or estimate the amount and concentration as good as we can. And that's enough for the toxicologist to go through and make a claim about genotoxicity, carcinogenicity, and so on. So this is the chemistry testing. If you had something like a permanent implant that includes a plastic and you can't rely on previous data, then you would go through this process. So I also want to take a second to talk about 18562 testing. This is another big change. It's another type of chemistry testing. ISO 18562 is one of these standards. It's relatively new, that for somebody like me and also Thor, anybody that's been involved with biocompatibility, we were just like waiting and hoping for something like this. And it was really great when it came out, because you can imagine for a gas path device, you still need to evaluate the biological risks just like you would any other device. But the testing strategies recommended in 10993-1 just really were not very appropriate for gas path devices. So for example, one that we would commonly see before is something like an oxygen concentrator, so like a portable oxygen concentrator. And depending on how familiar you are with these are like little machines that take air and compress it with an artificial zeolite. The zeolite absorbs selectively nitrogen, and then it releases oxygen-enriched gas to the patient. But if you take one of these machines and try to test it per 10993-1, where you're like wanting to soak the machine inside like a liquid and then apply it to the animal, well, this zeolite explodes in contact with water, and that's just, you can see it's just not going to work. So this is like really, really common sense for gas path devices. If you have a gas path device, you really must test to 18562. Now, the regulators aren't accepting anything, outside of 18562. If you do 10993, they'll be like, great, now go back and do 18562. So it's really common sense. I think it makes, I love it from a scientific perspective. If you have a gas path device, the risk that it has is really whatever it can carry in the gas to the patient. So gas passes through the device, it touches the patient's lungs. What can be inside the gas? Volatiles and particulates. So if your device is one of these where that's its only contact, then really you just need to measure the volatile organic compounds that can be carried from the device and check if it can release any particulates, and have those evaluated for safety, and then you're done. If you have a device, something like a breathing circuit where water can condense inside and then that liquid can run into the patient, that's kind of a separate thing. Then you need to understand what could leach into that liquid that the patient could swallow. So in that case, you end up doing extractables and leachables chemistry testing like I described before. The way that we run this is something that we have running inside Nelson Labs now, is to measure the volatiles. We direct clean filtered air through the device in a controlled environment. And then we use one of these vacuum canisters to sample the gas over a period of time. And then we can pump everything out of the canister into a GCMS and we analyze what's there that way. For the particulates, you can either use, the standard gives two options. You can either use a filter and measure it by weight, which is a strategy that we've done before, or you can use dynamic light scattering. So you have a laser across the gas path, and depending on the size and number of particles that the laser scatters more or less, you can measure the amount of particulates that way. So this is what you should do for gas path devices. If you're going to be submitting in Europe, you're going to need to comply with this. Okay, and then finally, at the end, after you made the plan and done the testing and the evaluations, I should really lead with evaluations and, if necessary, testing. Then you have the biological evaluation report. And like I said, this is a document that you may or may not have been used to preparing, but you sum up all the evaluations that were completed. And then there should be some conclusion there where, it's going to be signed by an expert or somebody qualified to make this claim. Like, okay, this device, it was evaluated for compliance. Everything passed. All the evaluations turned out good. Therefore, the device is biocompatible. That makes it really easy for your reviewer to go through and understand your point of view as you do this. Okay, so that's it. I would be happy to answer any questions that you might have. If you have questions that you maybe don't want to raise in front of the whole group, you're welcome to come and chat with us at our booth upstairs. Okay, I see a lot of very, very thoughtful but kind of shy people. So, okay, cool. Thanks for your time.